Good morning. Today in the 12th class biology, we have to study regulation of gene expression. We know that gene is the piece of DNA, and a piece of DNA have a character, and their regulation and their expression takes place through different pathways. And in that, what the different events are processed are transcriptional events. The first, transcriptional level. Second, processing level means if the introns are there and exons are there on the mRNA strand, then introns are spliced out, removed. And the second is after the mRNA formation, that mRNA is transported from nucleus to the cytoplasm to the ribosome. And in the ribosome, there is the conversion of languages of the nitrogenous bases on the mRNA strand in the form of polypeptide chain, that is called translation. After this, we have to study here that a gene is expressed, a gene expresses its character when it is required. When the particular induction, a particular character, a particular environmental condition stimulates the gene, guides the gene to be expressed, then only the expression of the gene is performed. It is said that here in the line that the expression is genetic expression is a metabolic, physiological, and environmental condition that guides or regulates the expression of gene. Means though all these metabolic, physiological, and environmental conditions are found in our surrounding or in the body, and that will be guiding the process of expression of the gene. Suppose lactose is a disaccharide sugar, and if lactose is a disaccharide sugar which is present inside the E. coli body, then E. coli have the need to form the beta galactosidase enzyme so that beta galactosidase can break the lactose and can form the two monosaccharides, galactose and glucose. And this glucose can be broken and energy can be emitted, and energy can be used for the metabolic and the life processes, regulation of life processes. Here we are looking that lactose when getting entered inside the cell, then only there is need of formation of beta galactosidase. Otherwise, beta galactosidase is not formed. But we have to know that the formation of beta galactosidase is dependent on the expression of a gene. And we have to know that the expression of gene is also dependent by the coordination of another protein, and that another protein that is known as regulatory protein. This regulatory protein will guide whom? The RNA polymerase. This regulatory protein will guide the RNA polymerase to perform the function. Okay, in this way, this regulatory protein, if guiding the RNA polymerase to bind at the promoter site and start the transcription, and not stop the transcription, that regulatory protein is actually expressed by a genetic sequence on the DNA strand, and that genetic sequence is termed as what is operator. This operator is the genetic sequence which has the duty and which has the responsibility to form that protein, which is the regulatory protein of the RNA polymerase, where that will be allowing the RNA polymerase to bind at the promoter site and start the transcription. So it can act in two ways. It can activate the process, activator or positively, and it can do the negative work, means it can repress the process. Okay, now then we are going to the case of lac-operon, which was discovered by the Francis Jacob and the Jacob Monod. These were the two scientists who said that the transcription of the DNA strand takes place in a particular metabolic, physiological, and environmental regulation. And in this way, he studied here that when the E. coli is ingesting the lactose as the food material, then there is the need of the formation of beta galactosidase, permease, and transacetylase. Three enzymes are needed so that the physiological process and the metabolical breakdown of the lactose can be done, so that the energy can be emitted and the life processes can be circulated and regulated. So that sequence of DNA, as it is the polycystonic sequence, because we know that bacteria E. coli is the prokaryotic, and in prokaryotic polycystonic cystone is formed, means the many polypeptide chains can be formed by the single structural gene, and these structural genes are regulated on the particular genetic sequence that is called operon. And in this way we know that the different structural genes which are found on the DNA strand, that which are Z, Y and A. These Z, Y and A are meant for the formation of different types of enzymes. These Z, Y and A are meant for the formation of different types of enzymes. And we have to know that this Z, Y and A will be expressing their character in the presence of or absence of inducer. Means this were doing their work in the presence of inducer, that is the lactose. And if the lactose is absent, then this all the enzyme formation will not take place. So, firstly, what are these works? The beta galactosidase, which is expression of the Z gene, its work is to break down or hydrolyze the lactose into the glucose and galactose. Here it is written. Second is, what is the function of Y gene? Means it will be increasing the permeability of beta galactosides in the cell, and that is the transacetylase. It is expressed by the gene A. Now, in the figure, we have to see in the figure what is the figure saying, that you will be looking that there is a gene, and that is lac operon. As this genetic sequence is guided and regulated in the presence of lactose, so it is called lac operon. You know that this is the promoter site, and this is also the promoter site, and this is the operator, and this is the operator which is meant for the formation of a protein that is called regulatory protein, and that is the repressor protein. In the absence of inducer, what is happening? When the RNA polymerase is coming to bind at the promoter site, that will be transcribing the regulatory gene I, and this regulatory gene I is making the repressor mRNA, and that repressor mRNA is translating and making the regulatory protein that is called repressor, and that repressor, just in the absence of inducer or lactose, that is going and binding to the operator site and do not allow the RNA polymerase to do the transcription of ZYA, as the inducer is absent, as the lactose is absent. But when the E. coli feeds over the lactose, and when the lactose comes into the cell, then the lactose is present as inducer, and when the regulatory gene I will be making the repressor mRNA after the transcription process, and that will be making the repressor protein after the translation, then the repressor protein will be bound by the inducer lactose, and this repressor become inactive. Now RNA polymerase can work further, can transcribe further ZYA, and can make the lac mRNA, and Z gene will be making beta galactosidase, Y gene will make permease, and A gene will make transacetylase. In this way, now the lactose is present, that will be hydrolyzed into the galactose and glucose is broken, and that will be emitting the energy. So, as its function is dependent on the presence or absence of inducer, and that is called lactose or allolactose. Lactose and allolactose, these two are the disaccharide sugars which are present in the cell, and if present, play the role of inducer; if not present, play the role of inhibitor. As the lac operon regulation, in the last line it is saying that as the regulation of lac operon is performed the basis of on the negative feedback, on the negative regulation, in the absence repressor is formed, and in the presence repressor is not formed. That's why we can say that lac operon as a negative regulation. The regulation of lac operon by the repressor is known as negative regulation. Thank you.